now I am 6. I'm as clever as clever. So I think I'll be 6 now forever and ever. You know that little poem, it always sticks with me.
Oh, yeah.
My grandmother, Vera, she used to say it all the time. 6 was like the perfect age for her. And she lived like that, so much energy.
Right.
Wouldn't even let us call her grandma. Just Vera. She taught me to drive way too fast.
Huh. Sounds like quite a character.
She really was. But then, as she got older, it felt like that Vera, the vibrant one, she just wasn't there anymore, long before she actually passed.
Mm-hmm.
And it makes you think, doesn't it? Is that decline, that sort of fading away?
Yeah.
Is it just inevitable? Or maybe could we actually change how that happens?
That's exactly the big question we're diving into today. You shared some really interesting material with us, and we're going to explore this whole idea of aging itself. The mission really is to unpack this concept that maybe aging isn't this fixed thing we just have to accept, but more like a condition, something we can understand and maybe even, influence. We'll be leaning quite a bit on David Sinclair's book, Lifespan.
Okay, condition, not inevitability. That's a pretty big shift right there. So what's the plan? What journey are we taking through this stuff?
Well, first we'll look at how people have thought about aging over time, how that understanding has changed.
Right.
Then we'll really get into this newer idea, the information theory of aging. It's a really different way of looking at it.
Information theory. Okay.
And finally, we'll touch on some of the, frankly, pretty exciting possibilities that are coming up, not just for living longer, but maybe more importantly, living healthier for longer.
Yeah.
That health span idea.
Health span, right.
So yeah, get ready for maybe a few aha moments along the way.
I'm definitely feeling that already.
Mm.
Okay, let's jump right in then. This shifting view of aging. It's strange to think now, but people used to just die of old age, didn't they?
Exactly. It was just this sort of catch-all. And what's really striking is how different that is from modern medicine.
How so?
Well, over the last century or so, certainly in the West, there's been this huge push away from just saying old age. Now it's all about finding a specific cause.
Like a particular disease.
Precisely. The WHO's International Classification of Diseases, the ICD, really shows this. Back in 1893, it had, 161 headings?
Wow, that's not many.
Right. And today, it's over 14,000. It just shows this incredible focus on nailing down the exact what and how of why someone died.
Okay, so we get really specific about the immediate cause. But the sources you share make a really crucial point here, don't they? Aging itself is still the elephant in the room.
It absolutely is. While we're busy classifying all these diseases, cancer, heart disease, dementia, aging is still the single biggest underlying us factor for almost all of them.
More than lifestyle factors sometimes.
Often, yes. The numbers are pretty stark. Like smoking might increase your cancer risk, say, 5 times, but just being 50 years old, that can increase it 100 times.
100. Wow.
And by age 70, it could be 1,000 times higher. And you see similar patterns for heart disease, diabetes, Alzheimer's. The list just goes on.
It's kind of a paradox then. We fight the individual fires the diseases, which is obviously vital, but we maybe ignore the conditions that make the whole forest flammable in the first place.
That's a great analogy. And there's this widespread acceptance, isn't there? People just shrug and say, well, getting old, that's just life.
Yeah, it is what it is.
But why? Why do we accept it when the impact is so huge? Maybe it's just so deeply ingrained that it's natural, unstoppable.
Mm-hmm.
But when you consider the suffering, the cost, both personal and economic, tied to age-related decline, that acceptance starts to feel a bit unsatisfying.
It really does. Like just accepting your car will fall apart eventually instead of, doing the maintenance to keep it running well for longer.
So before this information theory came along, what were the prevailing ideas? Why did scientists think we aged?
Well, some of the earlier theories were quite neat, really. There was Kirkwood's disposable soma theory. Disposable soma.
Yeah, the idea is basically that our bodies have finite resources. So evolutionarily, there's a trade-off between putting energy into reproduction versus putting it into long-term body maintenance and repair.
Okay, so prioritize making copies over maintaining the original.
Pretty much. Pass on the genes, that's the main goal from evolution's perspective. After that, investing heavily in keeping the soma of the body going indefinitely is less critical.
Does that hold up in nature?
You see examples that seem to fit. Think about mice, short lifespan, reproduce like crazy. Compare that to some birds that live for decades but reproduce much more slowly.
Right.
Or those chameleons Sinclair mentions, on islands without predators. They evolved longer lifespans. Less pressure, maybe resources get reallocated.
Interesting. So the body sort of budgets its energy. What were the problems with these early ideas? What did they explain?
Well, one big challenge came with Seelard's mutation accumulation theory.
Okay, what was that?
That suggested aging was mostly down to errors, mutations building up in our DNA over time, just wear and tear on the genetic code.
Makes intuitive sense.
It does, but then cloning came along, and that kind of threw a wrench in the works.
How so?
Well, if aging is just accumulated DNA damage, then if you clone an animal from an older individual cell, that clone should basically be born old, right? It should have all those accumulated mutations already.
Ah, right. And did that happen? I remember Dolly the sheep.
Dolly was the big one, and she did have a shorter lifespan, died relatively young from a lung disease. So at first people thought, aha, see? But looking closer, she didn't really show widespread signs of accelerated aging in other ways. And more importantly, since Dolly, lots of other animals, goats, mice, cows, other sheep, have been cloned from adult cells and lived perfectly normal, healthy lifespans.
So the original blueprint, the DNA itself, wasn't necessarily degraded beyond repair.
Exactly. It suggested that simply accumulating mutations probably wasn't the whole story. Something else must be going on, something beyond just errors in the DNA sequence itself.
So these single-cause explanations started looking a bit too simple.
Yeah, biology is rarely that neat, is it? The thinking started shifting towards a more complex picture. Aging probably involves lots of interconnected things happening.
And that's where this new theory comes in.
That's where the information theory of aging steps up. It offers a really compelling framework for thinking about that complexity.
Okay, I'm intrigued. Information theory. Break it down for us. What's the core idea?
The fundamental concept is that our bodies operate using two kinds of information. There's the digital information.
Our DNA sequence, the genes.
Exactly. That's the blueprint, the hard-coded instructions. But then there's the analog information.
Analog.
Right.
Like an old vinyl record versus a CD.
That's a perfect analogy Sinclair uses. The CD is the digital DNA. The code is perfect. The vinyl record is like the epigenome.
The epigenome. Okay, what's that?
The epigenome is essentially the system that controls how that digital DNA code is read and used. It's about which genes are turned on or off in which cells at which times. Think of it like the software running on the genetic hardware.
Oh, okay.
These epigenetic marks, they can be inherited when cells divide, but they aren't changes to the DNA sequence itself. They're like chemical annotations or tags on the DNA and the proteins around it.
So, using your analogy, the CD itself is fine, the genetic code is intact.
Right.
But maybe the CD player gets dusty or scratched, the epigenome gets degraded.
Exactly. That's the core of the information theory. Aging isn't primarily caused by damage to the genetic code, the digital information. It's caused by a loss of the epigenetic information, the analog signal that tells the cell how to read that code correctly. The instructions get noisy, corrupted.
Like scratches on the DVD, as Sinclair puts it. The data is still there, but the player struggles to read it accurately.
Precisely. And this loss of accurate epigenetic information leads to cells malfunctioning, losing their identity, and ultimately contributes to the diseases and frailty we associate with aging.
Okay, that's a really different perspective. So how does this actually work? Who are the players involved in maintaining this epigenetic information?
A really key group of proteins involved are called sirtuins.
Sirtuins. I think I've heard of those.
Yeah, they've gotten a lot of attention. They were first found in yeast. The gene was called SIR2, hence the name. Mammals, including us, have 7 different sirtuins, SIRT1 through SIRT7.
And what do they do?
They're crucial epigenetic regulators. Think of them as guardians of the epigenome. One of their main jobs is to remove specific chemical tags called acetyl groups from histone proteins.
Histones are the proteins DNA wraps around, right?
Exactly. DNA is spooled around these histones. Adding or removing those acetyl tags changes how tightly the DNA is packed.
Ah, so that affects whether genes can be read or not.
Precisely. Tightly packed DNA means genes are generally off, silenced. Looser packing allows the cellular machinery to access the genes and turn them on. Sirtuins help control this packaging.
So they're like cellular librarians, making sure the right books are accessible and the wrong ones are kept locked away.
That's a good way to think about it. They manage gene expression, but they also have other critical jobs, like coordinating DNA repair when damage occurs, and playing roles in metabolism and stress resistance.
Okay, busy molecules.
Very busy. And here's a crucial point. To do their jobs, sirtuins need a cofactor molecule called NAD-nicotinamide adenine dinucleotide.
NAD, okay.
Think of NAD as the fuel for sirtuins. Without enough NAD, the sirtuins can't function properly. And guess what happens to our NAD levels as we get older?
Let me guess. They decline.
They decline
Yeah.
significantly. And this decline in NAD is thought to be a major reason why sirtuin activity decreases with age, leading to problems with epigenetic control and DNA repair.
So less fuel means the guardians can't do their jobs as well, and the epigenetic information starts to get messy.
That seems to be a key part of the picture. This messiness is what Sinclair calls epigenomic noise.
Like static on that analog record.
Exactly. Over time, due to things like DNA damage, inflammation, metabolic changes, the epigenetic landscape gets disrupted. Genes that should be silent in a particular cell type might get turned on. Genes that should be active might get silenced. Cells start to forget their original identity and function.
You mentioned yeast studies earlier. How do they show this noise concept?
In yeast, researchers noticed these little extra circles of DNA called ERCs building up as the yeast cells aged.
Okay.
They found that the yeast sirtuin, SIR2, was getting sort of distracted by these ERCs. It was spending its time trying to deal with them, binding to them.
Instead of doing its main job.
Right. It got pulled away from its normal role of silencing certain genes elsewhere in the genome. This led to a loss of that silencing, causing sterility and other aging-like changes in the yeast. It looked a bit like Werner syndrome in humans, which involves premature aging.
So the noise, the ERCs, basically overloaded the system and pulled the repair crew away from their regular duties.
That's the idea. And they confirmed this by intentionally causing DNA breaks elsewhere in the yeast genome. Again, they saw SIR2 rushing off to the damage site, neglecting its other silencing tasks. It really supported this idea of sirtuins being diverted by damage or noise.
Okay, that makes sense in yeast. But how do we know this applies to us, to complex mammals?
That's the million-dollar question, right?
Right.
Bridging that gap. Mammals have 7 sirtuins, with SIRT1, SIRT6, and SRT7 being particularly important for epigenome maintenance and DNA repair.
So more complex system.
Definitely. And to test if this epigenomic noise could cause aging in mammals, Sinclair's lab did this really ingenious experiment with what they called icy mice.
Icy mice, inducible changes to the epigenome, right?
Exactly. They engineered these mice so they could specifically introduce breaks in their DNA, essentially creating that noise, those scratches on the record, in a controlled way. Crucially, they did this without causing mutations in the genes themselves.
So just disrupting the epigenome, not the DNA code.
Correct. And they didn't mess with other factors often linked to aging, like telomere length, mitochondria, or stem cells. They isolated the effect of epigenetic disruption.
And what happened to these ICE mice?
The results were pretty stunning. Introducing this epigenetic noise accelerated aging dramatically.
Really? How?
The mice got older, faster, they lost weight, their fur grayed, they became frail, lost muscle strength, developed cataracts, bone loss,
Right.
all the hallmarks of aging, but much earlier than normal mice.
Wow. So just scrambling the epigenetic instructions was enough to trigger accelerated aging.
It really looked that way. It provided strong evidence that this loss of epigenetic information, this noise, could be a fundamental cause of mammalian aging, not just a consequence. It supports the information theory in a big way.
That is compelling. If the genes are okay, but messing with how they're read causes aging? Wow. Okay, so if this information loss is key, can we learn anything from creatures that seem to resist aging? The sources mentioned some.
Absolutely. Looking at organisms with exceptional longevity gives us fascinating clues. Take bristlecone pines.
The super old trees.
Yeah, some are nearly 5,000 years old, and they show what scientists call negligible senescence. Basically, they don't seem to decline biologically in the way most organisms do.
So a 4,000-year-old tree is functioning pretty much like a young one.
Astonishingly, yes. Studies comparing young and old ones find very few significant differences in things like their vascular function, nutrient transport, even the quality of their pollen and seeds. It's like they found a way to maintain that cellular information over millennia.
Mind-boggling. What else?
Then there's the hydra, that little freshwater polyp. It has incredible regenerative abilities. It seems able to constantly renew its cells, potentially resetting cellular age in the process.
Wow.
And maybe the most extreme example is the Greenland shark.
How long do they live?
Estimates suggest potentially over 500 years. They don't even reach sexual maturity until they're about 150. Right. Their whole aging process is just on a completely different time scale.
Yeah.
It really shows that aging isn't some universal fixed law. Nature has found ways around it, or at least ways to dramatically slow it down.
These examples really hammer home that aging might be more flexible than we assume. Okay, so if it is flexible, if it's potentially modifiable, What can we actually do about it? What interventions are being explored?
There's a whole range, actually, from lifestyle changes to quite high-tech approaches. Let's start with the basics, things you or I could potentially do. Exercise is a big one.
Always comes back to exercise.
Pretty much. Studies have linked regular physical activity with longer telomeres.
Those caps on the ends of chromosomes.
Yeah, they tend to shorten with age, and exercise seems to help maintain their length, which might be linked to slower cellular aging. But the benefits go way beyond telomeres, obviously.
Okay, what else? Diet seems huge in this field.
Definitely. Dietary restriction has a long history in aging research. Calorie restriction, cutting calories without malnutrition, was shown to extend lifespan in rats way back in the 1940s. More recently, there's a lot of focus on intermittent fasting or time-restricted eating, and also fasting mimicking diets.
Do those work in humans?
Human studies are showing benefits, yeah. Things like weight loss, lower body fat, better blood pressure, and lower levels of hormones like IGF-1, which is implicated in aging pathways.
And didn't the sources mention something about centenarians having genes that mimic fasting?
Yes, some studies, like Barzilai's work with Ashkenazi Jewish centenarians, found genetic variations linked to longevity that seem to mimic some effects of calorie restriction, like their bodies are naturally in a more longevity mode.
Interesting. Are there specific food components to watch?
Research is looking into restricting specific amino acids like methionine, which increased healthy lifespan in mice. Also, lowering intake of branched-chain amino acids, leucine, isoleucine, valine found in protein-rich foods, is being studied.
Okay, so diet is complex but potentially powerful. What about that cold exposure idea?
Yeah, hormesis, the idea that small stresses can be beneficial. Historically, there was a drug, dinitrophenol, that increased metabolism like cold exposure does, but it was really dangerous.
Right, don't try that at home.
Definitely not. Modern approaches are things like cold showers or ice baths. The idea is they might activate beneficial pathways, like producing brown fat, which is metabolically active. Still needs more research in humans, though.
Makes sense. And avoiding bad stuff.
Oh,
Minimizing damage.
absolutely critical. Protecting our DNA, that digital information, is fundamental. That means being aware of environmental toxins, things like certain chemicals and plastics, pesticides, some dyes, industrial pollutants.
Like PCBs, azo dyes.
Exactly. And also minimizing radiation exposure, even natural sources like radon gas in some homes. Just reducing the burden of damage helps the whole system.
Right, basic maintenance. Okay, let's move into the more pharmacological side. Rapamycin keeps coming up.
Rapamycin is fascinating, found in soil bacteria on Easter Island, of all places. Initially an antifungal, but then found to significantly extend lifespan and health span in mice by inhibiting a key cellular pathway called mTOR, which is involved in growth and metabolism.
Is it used in humans for aging?
Not specifically approved for aging, but a related drug, everolimus, showed promise in boosting flu vaccine response in older adults, which is linked to immune aging. So there's interest.
And metformin, the diabetes drug.
Yeah, metformin is huge in this space. It's derived from the French lilac plant and is a frontline diabetes treatment, but studies suggest it might mimic some effects of calorie restriction and have broader anti-aging potential.
So people are taking it off-label.
Some are, yes, hoping for longevity benefits. There's actually a large clinical trial called TAME Targeting Aging with metformin planned or underway to specifically test its effects on aging in non-diabetics.
Okay, that'll be one to watch. What about boosting NAD? You mentioned that earlier.
Right. NAD boosters like NR, nicotinamide riboside, and NMN, nicotinamide mononucleotide. Since NAD levels decline with age and sirtuins need NAD, the idea is that boosting NAD could restore sirtuin activity and combat aspects of aging.
And does it work?
Animal studies are promising. Sinclair mentions unpublished research suggesting NMN restored ovarian function in old mice, for instance. And he shares that anecdote about his father feeling much better after taking NMN and metformin.
That personal story does make it feel more real.
It does, but, more large-scale human data is still needed for definitive proof of anti-aging effects.
Of course. What about senolytics? Clearing out old cells.
Senolytics are another really hot area. The idea is to target and eliminate senescent cells.
The zombie cells.
Yeah, basically. They stop dividing but don't die, and they hang around secreting inflammatory stuff that damages surrounding tissue and contributes to aging.
So senolytics are drugs that kill just those cells.
That's the goal. Early human trials are happening for things like osteoarthritis and eye diseases like glaucoma, where senescent cells are thought to play a big role. If they work, it could be a way to periodically clean house at the cellular level.
Like a cellular detox. Interesting. And then there's a really futuristic stuff, targeting jumping genes, epigenetic reprogramming.
Yeah, we're getting to the cutting edge now. Those line 1 elements, the jumping genes, make up a lot of our DNA. They seem to get more active as we age, causing instability and inflammation.
And sirtuins are involved?
Seems like SIRT6 normally helps keep them quiet. In mice lacking SIRT6, these elements go wild, and the mice age faster. Interestingly, giving these mice antiretroviral drugs like HIV drugs actually extended their lifespan.
HIV drugs for aging, that's wild.
It suggests these jumping genes might be a target, and maybe NAD boosters could help too by supporting SIRT6 function.
Okay. And the reprogramming, that sounds like science fiction.
It almost is. Using those Yamanaka factors, OCTA4, SOX2, KLF4, sometimes CMARC, often called OSK researchers, were able to essentially rejuvenate cells. The most dramatic example Sinclair gives is restoring vision in old mice by delivering OSK via a harmless virus to their retinal cells.
They reversed age-related vision loss.
In those mice, yes. It suggests that the epigenetic noise can be wiped clean, that cells retain the memory of youth somehow, and we might be able to tap into that to reset cellular age.
That's incredible. The potential is enormous.
It really is. But there are huge questions about how to do it safely and controllably in humans. We need to understand those youth markers that guide the process. And of course, there are massive ethical considerations, especially around gene editing, like the Heijian-Kui case highlighted.
Absolutely. Okay, we've covered a ton of ground, from basic theories to these mind-bending technologies. If these things start working, even partially, what are the bigger implications for society?
Oh, they're potentially massive. We'd likely have to completely rethink what old age even means. How we categorize diseases might change if we see aging itself as the root cause.
And economically.
Huge potential impact. If people stay healthier for longer extending health span, they could remain productive longer, potentially easing the economic burden of aging populations. Goldman Sachs even noted this potential double whammy of aging societies. Fewer workers, more healthcare costs. Extending health span could flip that.
So, changes to retirement, careers.
Definitely. Maybe longer careers, multiple careers, more need for lifelong learning, those skill badicals Sinclair mentions. Family structures might shift too.
But there are downsides or challenges, right? What about the planet?
That's a crucial point raised in the material. If billions of people are living much longer, healthier lives, what does that mean for resource consumption, waste, environmental impact?
Yeah, Alex's perspective in the notes was important there. Longer lives could be tough on the planet if we don't change our habits.
Exactly. The argument isn't necessarily against longevity research, but it emphasizes that we must tackle consumption and environmental sustainability alongside it. It can't be one or the other.
And what about fairness, access? The Gattaca scenario.
That's the other huge ethical minefield. Will these therapies be available to everyone, or will they widen the gap between the rich and poor, creating a biological elite? We need serious societal conversations and regulations to ensure equitable access if and when these technologies mature.
So a real paradigm shift is needed, maybe even declaring aging itself a disease.
That's a controversial idea, but Sinclair argues for it. Why? Because classifying aging as a disease could unlock massive research funding and shift healthcare focus from treating individual late-stage diseases to preventing the underlying aging process.
More focus on prevention and maintenance.
Exactly. And it needs public funding for that basic research, not just private companies developing specific drugs. We need to insist on the right to be treated for aging, regardless of age or economic status.
The sources mentioned disparities already exist. Australia seems to do better.
Yeah, the point was that some countries, like Australia, achieve better health outcomes and rank higher on human capital indices, possibly due to more equitable healthcare access, suggesting it is possible to improve things system-wide.
And technology like biotracking could help personalize this.
Potentially, yes. Continuous monitoring biotracking could detect diseases much earlier, like the story of Suzanne finding her cancer early. This allows for personalized, preventative interventions rather than waiting for symptoms.
It's not just about new drugs, but also existing tools like vaccines.
Absolutely. Preventing infectious diseases like pneumonia or malaria already significantly extends lifespan globally. Advances in vaccine tech, like mRNA vaccines, could play an even bigger role going forward.
And maybe even replacing parts, like organ printing.
That's another frontier. 3D bioprinting of tissues and eventually complex organs could solve shortages and eliminate rejection issues. It's still developing, but the progress is remarkable.
Okay, so much to think about. As we wrap up this deep dive, what are the really crucial takeaways?
I think the biggest one is this shift in perspective. Aging might be driven more by this loss of epigenetic information than by irreversible damage to our genes. It's like losing the software, not necessarily the hardware breaking down completely.
And because it's information loss.
It might be fixable. The interventions we've discussed, from lifestyle tweaks like diet and exercise to NAD boosters, senolytics, maybe even epigenetic reprogramming someday, they all aim at restoring or protecting that information. It offers real hope for extending not just lifespan, but health span.
Extending those vibrant years like my grandmother Vera had.
Exactly. Living healthier for longer.
Okay, and that final provocative thought for everyone listening. If we really do gain these extra decades of healthy life, how will you use them? What becomes possible? And maybe more importantly, how do we ensure, as a society, that these advances benefit everyone and the planet we share?
It's a conversation that's really just getting started, isn't it?
It absolutely is.
And this field is moving incredibly fast. So, keep seeking out reliable information. Keep asking those critical questions. The future of aging isn't written yet. We're actually helping to write it right now.